Hello everyone, welcome to Mediverse Atlas. In today's video, we are going to study about coagulation of blood. Now, we all know that our blood is in liquid state in our blood vessels, right? Now, what happens when this blood vessel is cut or when a blood is collected in a container? Now, this blood, the fluid, the liquid nature of blood disappears into a solid jelly-like mass. So, the coagulation of blood is the process by which liquid blood is converted into solid jelly-like mass. This process it is known as coagulation of blood. Now there are certain factors which help in this conversion of this liquid to a solid jelly-like mass. That is the coagulation of blood. There are certain factors which help in the coagulation of blood or the blood coagulation. These factors are known as the clotting factors. So these clotting factors are mostly proteins, that is enzymes, which are present in our tissue fluids or the blood. And we must remember that these clotting factors are present in inactivated state. So these are present in inactivated state. These clotting factors must get activated for the coagulation of blood. Okay, so there we'll be looking into the what the clotting factors are. So the first factor is the fibrinogen. The second one is the prothrombin. The third one is the thromboplastin or the tissue factor. Fourth one, the important one, is the calcium. Fifth one is the labile factor. Sixth one, still we don't know the sixth clotting factor. The seventh one is the stable factor. Eighth one is the anti-hemophilic factor. Ninth one is the Christmas factor. 10th one is the Stewart-Prover factor. 11th one is the plasma thromboplastin antecedent. 12th one is the Hegeman factor or the contact factor. Next 13 is the fibrin stabilizing factor or the fibrinase. So these are the clotting factors which are present in our body fluids or which are present in our blood. Now let us study the mechanism of coagulation. Before studying the mechanism of coagulation, I need to tell you something about the enzyme cascade theory. Now what exactly this enzyme cascade theory is. Now we all know that the clotting factors are in inactivated state. Now these inactivated clotting factors must be activated for the process of clotting, right? Now let us take an example of a clotting factor 12. Now this clotting factor 12, first it is activated. Activated clotting factor is represented like this, with the letter A beside the Roman numeral. Okay, so this Roman numeral represents the clotting factor. Beside the Roman numeral, this A represents the activated clotting factor. Okay, now let us take an example of clotting factor 12. Now this clotting factor 12 is activated. Okay, now this is the activated clotting factor 12. Now in turn, this activated clotting factor, it activates another clotting factor 11, like this. Again this activates the clotting factor 9. Okay, again this activates the clotting factor 10. So this process, the one clotting factor activates the another clotting factor. Again the one clotting factor, it activates another clotting factor. So this process, the mechanism, the process, the stepwise activation of each clotting factor, it is known as the enzyme cascade theory. Okay, so this is the enzyme cascade theory. I hope you got what enzyme cascade theory is actually. Okay, now we look into the stages of blood coagulation. So there are totally 3 stages of blood coagulation. Stage 1, it is the formation of prothrombin activator. Stage 2, it is the conversion of prothrombin into thrombin. Stage 3, it is the conversion of fibrinogen into fibrin. Okay, so there are totally 3 stages of this. We will be understanding each stage in detail. Now stage 1, it is the formation of prothrombin activator. Now this formation of prothrombin activator occurs to 2 pathways. The first pathway is the intrinsic pathway, the second one is the extrinsic pathway, and we need to remember that these pathways takes place simultaneously. Okay, so 2 pathways are there, intrinsic pathway and extrinsic pathway. Now what exactly these intrinsic and extrinsic pathways are? Intrinsic pathway, the formation of prothrombin activator, it is initiated by platelets within the blood. Okay, we all know that the blood has platelets, right? Within the blood, platelets activate the intrinsic pathway. Whereas extrinsic pathway is initiated by the tissue thromboplastin, and this tissue thromboplastin, it is released by the injured tissues. Okay, now we all know, suppose think that this is a tissue, think that this has a tissue and this has a blood vessel which supplies blood to this tissue, and blood has platelets, right? Now when there is an injury, along with the damage of tissue, there will damage of endothelial vessel also, along the blood vessel also. Now thromboplastin is released by this tissue. Okay, whereas this, the platelets, this activates the intrinsic pathway, whereas this thromboplastin, it activates the extrinsic pathway. Now let us study the intrinsic and extrinsic pathways in detail. First we will be studying the intrinsic pathway. Stage 1, it is the formation of prothrombin activator, right? We'll study the stage 1 intrinsic pathway. So when there is endothelial damage, there is exposure of collagen, right? We have studied about this in the last video. If you have not seen, please check it out. I'll provide the link in the description. Now when there is endothelial damage, in the presence of kallikrein and HMW kininogen, the factor 12, it gets activated. Okay, so now this is the activated factor 12. Now according to enzyme cascade theory, it activates another clotting factor, right? This activated factor 12, it activates the factor 11 in the presence of HMW kininogen. Okay, see, activated factor 12, it activates inactivated factor 11 to activated in the presence of HMW kininogen. Okay, see, I hope you are getting my point. This activates this. Now this is activated, right? So factor 11 is activated. Now this factor 11, in the presence of calcium, it activates factor 9. Okay, now this activated factor 9, in the presence of factor 8 and calcium, it activates factor 10. Okay, now keep this aside. This is one aspect. Now the factor 10 is activated. Now what happens is, when there is endothelial damage, there is collagen exposure also, right? Now these platelets are exposed to collagen. These platelets, when exposed to collagen, they release the phospholipids. Now along with the released phospholipids, activated factor 10, factor 5, prothrombin activator is formed. I hope you are getting my point. Please try to understand. See, factor 10 is activated by enzyme cascade theory, enzyme cascade method, or enzyme cascade, the stepwise activation of clotting factors. So factor 10 is activated. Along this activated factor 10, factor 5, phospholipids, these form the prothrombin activator in the intrinsic pathway. Now what is this phospholipid? It is released by the platelets when these platelets are exposed to collagen. I hope you are getting my point clearly. Factor 10, factor 5, phospholipid forms the prothrombin activator in the intrinsic pathway. Now let us see the extrinsic pathway. Extrinsic pathway, there is release of tissue thromboplastin, right? This tissue thromboplastin, it is composed of glycoprotein and phospholipid. Okay, now this tissue thromboplastin, in the presence of factor 7, it activates factor 10. Now this activated factor 10, in the presence of calcium, factor 5, and the thromboplastin. Okay, and the thromboplastin, it activates, or it forms the prothrombin activator. I hope you are clear about the intrinsic and extrinsic pathway. If you have any doubts, please do comment in the comment section. I'll be replying. See, tissue thromboplastin, factor 5 and calcium along with factor 10 forms the prothrombin activator. Now the stage 1 is completed. The prothrombin activator is formed. Okay, now let us study about the stage 2. Now in stage 2, prothrombin activator, it helps in the conversion of prothrombin into thrombin. So prothrombin activator, in the presence of calcium, it converts prothrombin into thrombin. Now what is this role of thrombin? First, it converts the fibrinogen to fibrin. This happens in the stage 3. We'll be studying. Okay, the conversion of fibrinogen to fibrin takes place with the help of thrombin, and this is the most important thing, positive feedback role of thrombin. Now what does the positive feedback role of thrombin indicates, or what does it signifies? Now see, look here. See, thrombin is formed with the help of prothrombin activator in stage 2. Now the formed thrombin, it sends the positive feedback control. Okay, it has positive feedback mechanism in the formation of prothrombin activator. That is the formation of thrombin helps in the formation of prothrombin activator also. In short, in the formation of prothrombin activator in the intrinsic pathway, factor 10, factor 5, phospholipids along with thrombin also helps in the formation of prothrombin activator. That is, the thrombin formed has a positive feedback mechanism or the positive feedback control in the formation of prothrombin activator. Even here, thrombin has a positive feedback control in the formation of prothrombin activator. So this is the significance or the most important point of the stage 2. See here, it accelerates the rate of formation of prothrombin activator. Which one? Thrombin, with the help of positive feedback mechanism. And also the thrombin, it activates the protein C. Protein C is an anticoagulant. We will be studying this about the protein C more in the topic anticoagulants. Okay, so just remember, it also activates protein C, which is an anticoagulant. Now the stage 3. Stage 3 is the conversion of fibrinogen to fibrin. Now, this stage 3 involves three main reactions. The first one is the proteolysis, second one is the polymerization, and the third one is the stabilization. Okay, so first here fibrinogen is formed, right? Look, thrombin, the fibrinogen which is present in our blood already. So this fibrinogen undergoes proteolysis and forms the fibrin monomer. Okay, this fibrinogen undergoes proteolysis and forms the fibrin monomer, and this fibrin monomer on polymerization forms the fibrin polymer. Okay, now fibrin monomer on polymerization forms the fibrin polymer. Now this fibrin polymer, with the help of calcium, factor 3 and factor 13, they form fibrin. Okay, factor 13 is fibrinase, right? And this process is only known as stabilization. Okay, so this process, it is known as stabilization. Okay, proteolysis, polymerization and stabilization. Okay, I hope you are clear. Fibrinogen is converted to fibrin monomer with the help of proteolysis. Fibrin monomer is converted into fibrin polymer with the help of polymerization, and fibrin polymer is converted into fibrin, that is the tough blood clot, into with the help of stabilization in the presence of calcium, factor 3 and factor 13, that is fibrinase. Okay, so this is the formation of the blood clot. Okay, see, you can see in this flow chart also. See, stage 3, fibrinogen A, that is fibrinogen, simple fibrinogen is converted into fibrinogen A, that is fibrinogen monomer. This process is proteolysis, right? And this fibrin monomer, it is converted into fibrin polymer, fibrinogen polymer. Monomer is converted into polymer with the help of polymerization, and this with the help of stabilization, it converts into tight blood clot. I hope you all are clear with this concept. If you have any doubts, please do comment. I hope you understood, and thanks for watching this video. See you. Bye.